Before you guys get started, we're going to talk a little about why we're here, remind you why we're here, what we hope to accomplish, why we go through this exercise, what you should take away from it. And one thing I think a lot of presenters but they overlook when they get into a room is not having a clear understanding of who their audience is. Okay? So we're going through the formality of introducing you to your audience so that you know where to pitch your presentation. You know, if you understand, if you're in a room full of neuroscientists and biochemists, you want to pitch your presentation at this angle. If you're a room full of early childhood education majors, you might want to pitch your presentation at a different angle because they don't want to understand the technology and the terminology, et cetera, et cetera.
And if you have Frank and Kristen, you might want to. I have to speak up.
Well, now we're going to make it really tough on these guys. Okay, I know where your mentor stands. So it might seem like formality to you all, but it's part of the process. Every time you give a presentation, you should really know ahead of time who you're speaking to, so you can craft your presentation accordingly. And also, this is probably the first time for this topic with this group that you guys are going to get some significant feedback. Okay, this is the point in time where you're going from all the preparatory stuff of the lit review, putting your thoughts together, organizing a concise presentation, to after today you're launching into it. I mean, jumping both feet into it, you should be running full steam ahead. Everything you get as feedback, what might be a criticism, might be a comment, might be a suggestion, is all meant to make sure that you have a much better product 2 years from now than you have now. And honest, we don't really care what your product is today. We know it's going to get better, okay? We just need to know that you're on the path. And everything you get back should be saying a way to improve where you are now to get to the next step. And the goal is 2 years from now you have a wonderful thesis, a wonderful publication that comes out of this process. So take it with that spirit, and that's what it's all about from here on out, okay, the next 2 years. All right, so now with the introductions, I'm Frank Cole, I'm director of the Gemstone program. I have a very, I think, solid fundamental understanding of basic biology, but neurobiology and biochemistry is not my forte. So you know how I'm going to be approaching my review of this.
I'm Kristen Scandal, Associate Director of the Gemstone program, and I've been taking biology in 20 years, but I think I understand what you're talking about. So.
Sir?
My name is Brian Beckett, Dr. B. And you know me.
Okay.
I'm Kara Duffy, and I am a neurobiologist by training, so here we go.
All right. And with that all being understood, Team Brain Blast is all yours.
Okay. Thank you, everyone, and thank you for coming to Team Brain Blast Gemstone Thesis Proposal Defense. I'm Caroline McHugh. This is Ashley Zachary and Shannon Morgan, and we'll be your presenters tonight. Our proposal is investigating the diabetic brain, the effects of thiazolidinediones and insulin on Alzheimer's disease. Okay, before we begin, I'd like to give you a brief introduction to what we're going to be talking about tonight. I'll begin, give you some background on Alzheimer's disease, what it is, and why it merits research, and how we're going to fit into that larger picture. Then I'll hand it over to Ashley, and she'll take you through what consists of our literature review and the current research being done. Then finally, we'll take it over to Shannon. She'll take us through what our study actually plans to do and the things we're going to look at and the data we plan to collect. Finally, we'll conclude with a summary of our budget and timeline for the next 2.5 years. All right, let's get started. Okay, Alzheimer's disease has had a substantial effect on society. Looking at monetary costs, in 2012 alone, the costs were over $200 billion to individuals in the government. Alzheimer's disease is the 6th leading cause of death nationwide. I'm sure many of you know those who've been affected by Alzheimer's disease. Or, in fact, the personal connection that some of our team members have is what led us to this line of study. What's more, Alzheimer's disease is the only one of the top 10 deadly diseases that has no cure, and its cause still remains unknown. Clearly, Alzheimer's disease is an area that merits further research. Okay, so we know Alzheimer's disease is important to study and it's bad, but what is Alzheimer's disease? Well, it is the most common form of dementia. This disease causes neurodegeneration, which means cells die through progressive apoptosis. In addition, neural pathways in the brain and connections are broken down through this disease. And as you can see here, in this comparison of a healthy brain with an Alzheimer's affected brain, the brain mass itself shrinks dramatically. And you see these large holes or vacuoles in the brain that are not present in the healthy brain. Alzheimer's disease is only officially diagnosable in an autopsy, where you look for Alzheimer's disease indicators such as plaques and tangles. These plaques and tangles are very important to understanding the pathology of Alzheimer's disease. So we're going to look at those a little closer. Again, we have a comparison of the normal brain with the Alzheimer's affected brain. There's two main differences you'll notice between them, and these relate to the plaques and the tangles. We'll take a look at the plaques first. The tangles, I'm sorry. In the normal healthy brain, tau proteins make up microtubules which transport essential nutrients in the cells. In the Alzheimer's disease pathology, this tau becomes phosphorylated. And this causes the microtubules to break down and form tangles. In addition, plaques form progressively throughout the disease, and these are not present in the healthy brain. These occur when APP, or amyloid precursor protein, is cleaved incorrectly, and these fragments of proteins that are left over bunch together in the brain. They clump together progressively, and they get bigger, and these things are found within the synapses, and they decrease the connectivity of the brain. So in conjunction with the tangles, this is what causes apoptosis. Okay, so in order to understand how Alzheimer's disease is researched and modeled, it's important to understand there are two types of Alzheimer's disease, familial and sporadic. The majority of cases of Alzheimer's disease are sporadic, which means the exact cause is unknown. In contrast, familial Alzheimer's disease is an autosomal dominant disease, so it's genetically linked. This is helpful for research because you'll know that your model will have Alzheimer's disease. So why do we use a mouse model? Well, mice's brains are similar to humans, and we can give them transgenes, which are human proteins put into the mouse from various loci, and those will ensure that the mouse has Alzheimer's disease in a way that is comparable with humans. So we can study it, and that's what we plan to do in our study. There's two main things you can do with an animal model. You can better understand the pathology of the disease, and you can test potential treatments or therapies. What we aim to do is a little bit of a combination of the both. The specific mouse model that we plan to use is the triple transgenic mouse. As the name suggests, there are 3 transgenes in this mouse from 3 different loci in the human genome that we put into the mouse to ensure it will definitely have Alzheimer's disease, and that's what we need in our study. This mouse will have onset of this disease at about 1 year of age, making it perfect for our study. All right, now I'm going to pass it over to Ashley, and Ashley will take you through the current research in the field and how we fall into that.
Thank you. All right, so currently there are a lot of different types of research surrounding Alzheimer's disease. Some focus on slowing and delaying the progression of the disease, some with identifying early warning signs in humans, and others are interested in understanding the pathology, how these amyloid beta plaques and p-tau tangles are getting there in the first place. Our research is going to fall under this category because we're interested in investigating what part of a signaling pathway may contribute to the pathology. Now, in addition to the plaques and tangles, decreased glucose metabolism is also associated with Alzheimer's disease, giving it the name type 3 diabetes, as some articles like to call it. We've been interested in looking at the literature that establishes a connection between diabetes and Alzheimer's disease, such as the study done in 2012 by Bonfin et al. In this study, insulin was used to investigate the amount of plaques and p-tau tangles in an Alzheimer's brain. Insulin is known to be a competitive inhibitor to the APP protein, which incorrectly cleaves and causes the amyloid beta plaques. When increasing insulin levels in the brain, it was found that the amyloid beta plaques and p-tau tangles were decreased, but the mechanism was unknown. Other studies have been investigating diabetes medications, such as thiazolidinediones, which I'll go into now. These are diabetes treatment and are the #1 most popular right now. As some of you may know, Avandia and Actos are currently advertised on TV. Now, in these studies, the thiazolidine diodes were introduced into the mouse, and amyloid beta plaques and tangles were also seen to decrease. Again, the mechanism is unknown. This is a big hole in the literature. We still don't know what is causing these plaques to happen. In these studies, TZDs were given orally because, unfortunately, they do not easily pass the blood-brain barrier. This is a chemical structure of pioglitazone, the TZD we will be using in our study. Even though it has this lipophilic side chain, which means it's dissolvable in fats and lipids and should be able to cross a membrane, it is large and is seen as a toxin, which is why not many molecules can pass through the brain. This is a problem because we want to see how TZDs are going to directly affect the brain, which is why we're going to propose an intranasal method. In the studies that used insulin to investigate Alzheimer's disease, insulin was given in a new intranasal delivery system. In this system, as I will show using this diagram of a mouse nasal cavity, the insulin was squirted into the nose and was absorbed through the olfactory epithelium. The olfactory epithelium has pathways that go directly to the brain, thus bypassing the blood-brain barrier. In this way, they were able to get the insulin to the brain without having to go through the entire bloodstream first, causing peripheral effects. Our team proposes to do the same thing with TZDs, in addition to using intranasal insulin. And we're going to use a combination of these to study where in the insulin signaling pathway Alzheimer's disease might occur. As you can see in this diagram, the insulin signaling pathway is very large. This isn't even a full diagram of it. There are a lot of different components, but our team will only be focusing on this part of the disease. In this part of the pathway. In this part of the pathway, insulin binds to the receptor and recruits these other components in the cell to send a message to this nuclear receptor called PPAR gamma. Now, PPAR gamma is important because it increases insulin sensitivity, and as I said before, one of the problems with Alzheimer's disease is glucose metabolism and insulin insensitivity. It also helps glucose flow into the cell, and glucose is important for cells to grow, function, and divide. Using a combination of both insulin and TZDs, we will be able to activate PPAR gamma because TZDs are PPAR gamma agonist. We will be approaching it basically from both directions, activating it from using insulin and TZDs, and by doing this, we hope to see an even decreased amount of pathology in the brain, which leads me to our hypothesis. We hypothesize that intranasal TZD will have a long-term additive effect to the intranasal insulin treatment, therefore improving the glucose uptake and decreasing the pathology in the brain. This would then suggest that a problem in the downstream insulin signaling pathway mechanisms are responsible for the pathology and would warrant future research by other scientists. Our research questions are then, are intranasal TZDs and insulin synergistic? Are they decreasing the pathology and increasing the glucose uptake more than a control? And also, following 8 weeks of intensive treatment with the TZDs and insulin, are these effects long-term? Are they still going to be there? And with that, I'm going to pass it over to Shannon, who's going to tell you how exactly we're going to answer these questions.
The first part of our study will be a pilot study, which we will be testing how long the effects of the TZD intranasal treatment stay in the brain, and we will use this to determine the dosage for our main study. As you can see here in this diagram, we will start with 6 non-transgenic mice, which means they do not have these transgenes for Alzheimer's disease. We will then give them an intranasal treatment of both TZD and insulin, here at time 0. We will split them up into 3 groups, 1, 2, and 3, with 2 mice in each group, and sacrifice and analyze the mice 1, 2, and 4 days, respectively, after the end of treatment. We will use this analysis of how long we can see the TZD in the brain to cut down on the number of times we have to treat the mice with TZD in our main study. As it is an 8-week intensive treatment, as consistent with the literature, we would have to normally anesthetize the mice to give them the intranasal treatment, and this would happen once a day. But our anesthetic, isoflurane, is very bad for the mice's health over a long-term treatment, such as 8 weeks. So if we can use a time other than 1 day, we can cut down on the number of times we anesthetize the mice and use that time for our main study, which you can see here. Our main study will hopefully indicate that some part of the downstream insulin signaling pathway mechanism contributes to Alzheimer's disease pathology. To determine this, we will be using our triple transgenic mice, and intranasal insulin, TZD, compared to control of intranasal acidic saline and insulin to see if we can see any difference in the pathology, either in the brain or in the behavior. Some things that we'll be looking for are their cognitive function. We hope to see it improved, as this is an outward effect of how the pathology in the brain is going. We will also be looking at the major hallmarks of Alzheimer's disease, such as amyloid beta plaques and p-tau tangles. We hope to see these decrease, as well as apoptosis, which, as we said, is the end result of Alzheimer's disease, cell death in the brain. We're also looking for some measurements to be increased. We're looking for increased neuron proliferation. This should indicate that not only is the brain not unhealthy with Alzheimer's, but that it's showing signs of health by giving birth to new cells. We'll also be looking for increased glucose uptake, as one of the major problems with Alzheimer's disease is not enough glucose in the brain. We finally will be looking to hopefully have no peripheral effects with this TZD treatment, which means that the intranasal TZD is going into the central nervous system, staying there and not affecting the periphery of the body. Our study will be very novel, mostly for the fact that the intranasal method has never been applied to TZDs before. This means that anytime a study has been done with TZD of any kind, it was administered orally and stays mainly in the periphery of the body. As Ashley already said, TZD is large and does not easily pass the blood-brain barrier. By putting these TZDs directly into the brain and the central nervous system using this intranasal method, we should see some differences in our study with other studies, as the main place where Alzheimer's pathology takes place is the brain. Our study is also novel because we'll be looking at a long-term measurement of Alzheimer's disease treatment compared to other studies, which you can see here in our flowchart of how our groups will be assigned. We start here at the very top with 45 triple transgenic mice. All will receive an 8-week intensive treatment. They're split up in colors, group 1, 2, and 3, depending on what kind of treatment they will get. Group 1 is our control group. They will be giving intranasal acidic saline. Group 2, intranasal insulin, and Group 3, intranasal insulin and intranasal TZD. Consistent with the literature, we will start by sacrificing 1/3 of our mice in this group, Group A on this line, 36 hours after the end of treatment. This should show us how we compare to the other literature. But as I said, our test is novel, so we will also be waiting to sacrifice 2/3 of our mice, 1/3 at a time, 2 weeks and 8 weeks respectively after the end of treatment. This will show us if the 8-week intensive treatment has a long-term effect compared to only 36 hours after testing. In addition, these B and C groups here, we will also be using cognitive testing 1 week before they are sacrificed to see any outward effects of the AD pathology in the brain. Some data collection that we will be using for behavioral tests, we will be using the Morse water maze, which is a large tub of water, and you place, to put it simply, we track the mice around the pool and test their timing to see their cognitive function and their memory, which, as I said, is the outward part of their AD pathology. Most importantly, we'll be doing a large number of postmortem tests on the mouse brains after their sacrifice. Metabolomics is a study of metabolites in the brain, and metabolites, if you don't know, are the products and intermediates of metabolism. ELISA assays will be using antibody and antigens immunostaining of the brain slices, and this will show us the major hallmarks of the disease, shown here on the left of amyloid beta plaques, and here p-tau tangles. Promegoluminescence shows caspase activity in the brain as well as apoptosis, so we will see if these brain cells are still infected, well, not infected, have Alzheimer's disease and die eventually. BRDU staining shows us neuron proliferation in the brain, so it shows if the brain is healthy and regenerating and the nerves are keeping born. And TZD in the periphery of the body has been shown to have the effect of weight gain on mice, so we'll be using inguinal fat dissection as our last measurement to see if the TZDs are affecting the mice in the periphery or not. Hopefully they will not, and we can still see that the TZDs are in the CNS and in the brain and staying there. We are lucky enough to have a fantastic mentor and expert who are providing us with lots of our resources, such as our mice, mass spec, isoflurane, and the Morse water maze. However, our project still will have a large number of costs, mainly, as you can see up here at the top, mouse maintenance and postmortem tests. We estimate that our project will cost us about $8,700 over the next 2.5 years. The next 2.5 years are shown here in our team timeline. Most notably, we hope to start pilot testing at the end of this spring semester, 2014, and be finished by fall of 2014. This is also when our mouse colony will be of 1 year of age. As we said, that is the onset of diabetes. So we can start our main study testing then, and hopefully be done data collection by spring of 2015. From spring of 2015 until our team's graduation in 2016, we'll be working on completing our thesis, presenting at conferences, and eventually end with the senior thesis conference in spring of 2016. However, our successful thesis would not be at all possible without our fantastic mentor, Dr. Burkett, our expert, Dr. Kara Duffy, our librarian, Natalina, our Gemstone directors, Dr. Skendel and Dr. Cole, our patent librarian, Jim Miller, and Dr. Angela Black. And with that, we'd like to open the floor up for questions.
Well, very good job. A very nice, strong presentation. Great pitch. Nice presentation. We typically start our questions with our outside expert first.
Okay.
So in order for Kara to get her outside, right? The biggest, most serious question I ask you, and this one you gotta get right. How do you pronounce your librarian's last name?
Changalova, I believe.
Say it again.
Changalova.
Changalova. Okay. That wasn't a very
Can I
serious
ask a
question.
quick methodology clarifying question?
That's all it takes.
For group C, are you doing behavioral testing twice, like once with group B and once before sacrifice, or just?
No, we're just going to do it once because we would assume that they would have the same testing abilities as group B because they were the same up until that point. We just test them longer.
Okay.
Okay, so Carol, let you go first.
Okay, I guess first, I would like to commend you guys on a well thought out proposal plan. It makes sense to me. It's very ambitious, but I'm impressed by that, so.
Yeah, you guys really, not that you hadn't pulled it together, but everything came together. I could see that.
So, I guess I would just kind of throw out an easy question to start with. Can you kind of explain a little bit how dementia differs from Alzheimer's disease?
Okay, so dementia is, in general, cognitive decline, but Alzheimer's disease is a specific form of that. And so I guess the difference is what causes that cognitive decline. So in Alzheimer's disease, like we said, the tau tangles and amyloid plaques versus other types of dementia, such as the second most common, Parkinson's disease. Although they're similar in many ways, and in fact, some research actually looks at the connections between the two. Well, that's not what we're looking at, but it also is very interesting that a lot of forms of dementia have a lot of similarities.
So not only are the pathologies different, but also the parts of the brain that are affected are also very different, right?
Yes.
So in Alzheimer's disease, what are the most important parts that are affected?
A lot of parts in the brain really surrounds like memory, hippocampus. I know from personal experience, having a family member with Alzheimer's disease, the memory fades, you can't recognize faces, family members, and you can also start forgetting how it affects parts of the brains that store procedural memory, how you would do a task that you used to be able to do and now don't remember how to do. And other forms of dementia can affect different parts of the brain, sometimes the cortex and personality, but in Alzheimer's disease, a lot of it's the memory that gets affected.
And in the very final stages, they often become bedridden and incontinent, so you really just can't do anything yourself.
Yep, you rely on other people. Okay. So another thing I noticed reading through your proposal is that a lot of the proposed rationales for using TZDs, and specifically when considering Alzheimer's disease, look at effects on insulin, APP, like you mentioned, inflammation. But did you consider, because of the effects on glucose metabolism and those sorts of things, looking at mitochondria? Because mitochondria are absolutely essential for energy, not only in the body but also in the brain. So is that something that you considered or would consider?
Certainly never came up before, but that
doesn't mean we're not open to it.
Yeah, it's just something to, a suggestion.
Yeah. Is there actually a change in the number of mitochondria?
Not necessarily the number, but they become less efficient. And there are, you can look at like oxygen respiration.
Do you test that when they're alive, or is that
No,
postmortem?
it would be postmortem, yeah.
Isolate mitochondria, yeah.
Just a thought, and you know, when you're looking through the literature too, it's, you know, something to be aware of.
Well, I'll add tag a question onto that. How might you be able to get some sort of an idea, it may not be mitochondrial, but in terms of metabolism in the brain? Who's the biochemistry people here? What occurs in the mitochondria compared to in the cytosol?
The energy like making ATP and for the
Right.
oxidative
Oxidative phosphorylation, TCA cycle. And that's something that metabolomics will be able to pick up.
Okay, let me tie on to that one from a somewhat naive point of view. You said that phosphorylation of in the axon causes the tangles?
It's phosphorylation of the tau protein, which makes up the microtubules.
So is
And
that tied to a mitochondrial,
No,
Okay.
no.
It's just when the,
similar term?
Right.
Okay.
when the
No,
tau,
not at all. Sorry.
when the tau becomes phosphorylated, it changes shape, and a lot of biology is about shape and where the charges are distributed. So phosphate is very negative, 3-, so that just changes the structure of the molecule and can really degrade everything and not be able
Okay,
to
so
function.
it's a physical deformation.
Yeah.
Yes.
Continue, please.
So I guess I was also gonna point out too, I mean, you guys talk a lot about glucose uptake, which is obviously because that's what you're focused on, but TZDs do a lot of other things too. They, in, you know, the same kind of vein, they have impacts on inflammation, they impact oxidative stress. These things are also very important in Alzheimer's disease. So how do you think that you can kind of separate these? You know, if you want to look more at glucose effects versus these other things that might be having an impact, how do you think you might tackle that?
I think using the metabolomics, we can look at the glucose pathway more specifically. I'm not wrong about that. So then we would be able to differentiate what's happening in that sort of pathway versus another pathway, which is like the advantage to using that.
Well, what about on the inflammatory side? You guys, who's been the one reading up more on that?
When you look at
What
people-
cytokines might, these are the ones that pro-inflammatory, might you look at? And there are methods to measure these.
Absolutely.
I guess that's something we haven't really considered yet, but
I
we will
remember
not
reading
consider.
a lot about PPAR and how the TZDs have inflammatory effects, anti-inflammatory
Right.
effects, and that inflammation is one of the parts contributing to Alzheimer's disease, but it wasn't necessarily what our project was focused on,
Right.
so
But I
it's
think
hard to
early on,
remember.
I mentioned that actually
Yeah.
what that might be, I think probably.
So I think even though it's not what you're focused on, you
Yeah.
just need to consider it because it could
Right.
still be having an effect on what you see. And so you just need to, you know, kind of squirrel that away in the back of your mind.
TNF alpha, IL-1 beta, I think, those
Oh,
are
yeah,
two
that was
most, and IL-6.
2 semesters ago.
That was a while.
See, that's my class.
Supposed to remember this thing.
I have notes, sorry. Okay, in general, with any study using like a transgenic mouse, I think it's a good idea to keep in mind the limitations of using that kind of approach. And while human and mice, you know, the genomes are probably like 80% similar, transgenic studies are a very powerful tool, but there are pretty big differences between a human brain and a mouse brain. So have you considered some of these things? For instance, a mouse brain has proportionally more neurons than a human brain does.
One thing that was interesting is that the olfactory epithelium of mouse is like infinitely larger than that of a human, so it was hard when it came to doing the dosages because we weren't sure what to use as like a proportional thing. So if you consider that a part of the brain, then that was definitely a confounding factor and makes our dosage calculation a little more less specific or less accurate. So that's one thing that we have run into.
I have to follow up, if I could. I have exactly the question I had written down about the dosage rates for the use of the mouse model and the transnasal effectiveness and relating that to the human. I mean, you can have great data for how it affects the mouse. Somewhere before you end the last of your thesis, you want to say how this translates to the human population. How do you go about making that leap or that transition to taking your data from your mouse? And I just now learned that they have a much more extensive nasal connection and relating that to the human population.
Well, do you want to take it?
You can start.
All right. Well, firstly, insulin is actually you can actually use human insulin in mice, so that'll make the transition a lot smoother to go from mouse to human.
Okay.
And also, there are a lot of tests being done already using intranasal insulin in humans, so that transition can be done very easily as well. But to answer your question regarding the transition between mice to TZD to humans to human intranasal TZD, that would be a much more difficult transition to make, and I think that in and of itself would be almost another study to conduct. So it's difficult for us to answer it at this stage.
If I could add on to that, because TCDs are not used intranasally and it's a relatively new method, there isn't a lot of information out there. So just for us to figure out the dosage calculations, which is extensive as you can see, we
I
did.
can see it's small.
This is just taken right from the proposal, but just to do based on the ratio of how big is the mouse brain compared to what the human brain would be. There's not a lot of research out there, even just in a mouse, so it really would warrant further studies.
So is the dosage really important?
We think that it would be better to give them too much than too little because the excess would just run off. So I think we were a little bit liberal in our calculation. We knew the ideal amount of volume of liquid to use for the mice, so we used that as our volume, and then in the calculations as it shows, we use other factors like glucose metabolism in the brain versus body, and then how much you'd actually give them in the body to convert it to how concentrated we should make that 24 microliters, which is max, like a good amount for the nose. So I forget what you said.
I just said is the rate really important? I think that's what I asked.
Well, kind of adding on to that, the method itself, like the mouse is held perpendicular and the drops are put into the mice's nostrils. So, like she was saying, runoff, most of it will go in, but it's kind of also accounting for the fact that some of it might not.
Mm-hmm.
And then I guess we're figuring out the rate of how much we have to give them by the pilot study. So.
I mean, exactly hit where I was going. I don't think rate matters as long as you have enough. You're trying to demonstrate that it will happen.
Mm-hmm.
Rate study is further down the road. I don't think rate really matters as long as you have enough.
That was on the insulin, as we learned with Team Pancreas.
We learn a lot. Okay, great. Great question.
But again, it's like I said at the very beginning, just to remind yourself, you said this, that TCD is both a treatment, but also a way of interrogating the pathway.
Okay.
And so that would answer your second part of your question. My second would answer your question.
I'm sure it does.
Yeah.
I carry one guy.
Okay. So again, I guess kind of taking a few steps back, keeping in mind some of the quirks of working with transgenic animals. Some of the earlier studies with some of the strains, you would see things like really robust effects on pathology but no memory deficit, or you would see memory deficit but no, less pathology. And that's something that happens a lot with these kinds of, strains and models. So is that something that, I guess I just want you to be aware of if you're not already, something also that you need to think about a little bit, just in case you see something in some of your measurements but not others, doesn't mean that your study is, invalidated. It happens. It's very common.
What's one thing you guys could do with statistically? And I don't know if we talked about this or not, but my previous team did, and they got a lot of value out of it. So you're making, especially in the 2 and the 8 week guys, well, even the 36-hour ones, you'll have pathology, but you won't have the behavior stuff. You're gonna have a lot of information on each mouse. And there could be things that you can try to correlate that would cause them to cluster. Has anybody heard of principal component analysis? Haven't had stats? You can use that to identify, as Kara said, ones that have gone more towards this pathology, or you don't see this, but it may actually relate to some things you're measuring. And that is gonna be valuable too. It's because it's almost like, prescribed, if you can come up with a measurement that will allow doctors to prescribe certain modalities, that would be very valuable too.
Okay. In some of your postmortem analysis testing, you were talking about some different methodologies that you would be using. And you weren't necessarily always very specific about where you would be looking in the brain, and that kind of goes back to what I was asking earlier about thinking about dementia versus, Alzheimer's disease versus Parkinson's disease. All of them have kind of specific target areas that are affected. And so with your postmortem testing, that's something that you can keep in mind to refine not just cost, because a lot of times you can run less samples if you're looking at only specific areas or smaller samples, but it also makes it more relevant to what you're trying to kind of ascertain. So
I guess we could use that to just homogenize the hippocampus when we're looking at the BRDU staining, because that's the only place of neurogenesis, so that's
Right.
good.
And then along the lines of that, I think that there is also some potential confusion methodology-wise. So if you can put it back up if you want. There's that picture actually, that one. This is immunohistochemistry staining. It's actually done in sections of tissue, versus the ELISA that you talked about is actually.
Different picture, I thought though.
So both of those,
Oh, okay.
it's the same thing, similar technique. So these are actually using, it's a completely different technique. It's very easy to do, but it is different in that this kind of technique tells you a spatial location where pathology is occurring, versus an ELISA, which is going to just homogenize a part of the brain. You could take something specific like hippocampus or a certain part of cortex, which then refines that part of the brain that you're looking at. But those are two separate procedures. And the only way I know how to do BRDU is with sections of tissue. I don't know that there's an ELISA available for something like that.
Yeah. Does anybody know what's the BRDU labeling method? Can you explain it?
I literally learned it today in my cell bio class, but it was at 9 in the morning.
It uses
It's gone already?
Yeah, bromodeoxyuridine, this is a thymidine analog,
Yeah.
and it labels up only what type of cells,
Proliferating?
right? And you visualize then the number of proliferating cells relative to the total number of cells to give you a proliferative index. And that you have to visualize like that.
So I think both techniques are valid and they're both, easily attainable for you guys, but there's just a, disconnect into what I guess you're actually doing.
What other measure of proliferation can you make other than using this thymidine analog? Anybody investigated other markers of cell proliferation?
Is it one with green and purple staining?
KI
67?
67? And nuclear origin?
I'm not sure
I think
offhand.
it is, yeah.
The benefit of using Ki-67 over BRDU is that BRDU requires that you inject the animals for probably like 5 to 7 days before you sacrifice them. KI-67 doesn't require anything like that. So if you wanted to look at a proliferative marker, you're potentially doing something less invasive to the animal right before. And it gets at the same point, so it's also doable. It would be also be like an antibody staining.
Are you going to provide these comments to the team written?
Yeah. I will type them into their Word.
Do you need them typed?
We'll have this recording,
You can just give them that. Unless you want to edit.
Unless you have comments. What
This is
the is this?
I guess another thing just to throw out there, which I know that we've talked about a little bit already, is that it will be good for you guys, both for kind of learning how a lot of these techniques work, to do some pilot testing with the actual transgenic mice as well, so that you get an idea of, you know, the techniques that you'll be using and how to run through a lot of that. And it wasn't mentioned in your proposal, but.
Do you mean the non-transgenic for the pilot? Like doing
Well,
the-
it's kind of additional pilot testing with
Oh,
the.
Because you
Oh,
said,
like the ones that we're sacrificing to figure
you
out.
said there are some transgenic mice of age now that we could use as
In
well.
like another month or two.
That would work
Or
perfectly.
some-
Practice mice.
Yeah, so you're working
Yeah,
with something
practice
you know
Yeah.
has, you know, PTAW.
We've learned about
tiny-
practice mice.
Would we need to have any kind of like approval
ICA?
to do that before? No.
They're just practice mice.
Just sack 'em.
So, you know, because I have a protocol, I can probably sack the mice and then just provide you guys with the tissue, so.
I hate that. It makes me so sad.
Very
They're
easy.
serving their purpose.
Yeah, it's a,
So,
Too soon.
in, thinking about the intranasal injection, you mentioned in the proposal that there are 2 pathways. What influences the pathway that either the insulin or the TZD goes? Is it random? Is it,
I believe that that both of the pathways will occur simultaneously. And I am rusty on what the specific names are of both of the pathways, but
The intraneuronal.
yeah, so essentially one will go directly into the brain, the other one will go from neuron to neuron. So the one that goes from neuron to neuron is much slower and is more like a delayed release, and then the other one is directly into the brain.
okay.
So they should occur, they're occurring simultaneously, but they won't reach the brain at the same point in time.
Do you think that matters for the effects that you're looking for, like 1, 2, 4 days later?
Well, a good amount will reach the brain through like within a couple of minutes. So I think if let's say it splits like 50-50 or something, we will be able to see like the effects that we're looking for. And then the rest, if it gets there through the other pathway, that'll have its added effects. But I think our main effect would be there.
Also, part of the reason that we did choose to do it at the different time intervals was to take that into account. So we don't know yet, but that's part of what we're testing.
maybe we need to leave your food though.
Okay, yeah, let me see.
You can stop whenever you want, okay?
Why don't we go ahead and move on to some
Okay.
other questions,
Yes.
and that way, because you can give your written questions
Yes,
to
I
them
can,
anyway.
yeah, absolutely.
But I really do, there's nice in-depth commentary there, that's very, very good.
Hey, guys, that's what I'm good
Yeah,
too.
not cutting you off, I hope.
No,
Okay.
I mean, I wrote down a lot of stuff, but
Oh,
I,
good, that's what we're
It's
really
lovely
thankful for
time.
you doing that, I mean, it's all meant to make it a better project. There you go. Could you flip, I'm gonna ask one, could you, want to ask one?
No,
Okay.
I ask them all the
Can
time.
you flip back to the slide in your methodology which showed your treatment groups, that one?
Yes.
Okay. Okay, convince me why you don't need to have a TZD group by itself. Why don't you need a
Because
4th group?
TZD, or pioglitazone, which we'll be using, is a enhancer for the downstream insulin signaling pathway. So if we just use TZD and no extra insulin, it's possible, correct me if I'm wrong, that we could just be seeing the same amount as if it was just insulin.
Also cost and the amount of mice we'd have to add on an additional 15 mice, and then pay for more of the drug. We just decided we originally had that and then decided to cut back.
Okay, so as an outside critique, looking at just the separate experiment, are you going to be able to explain to me the benefit of TZD, or are you going to be able to explain to me that TZD alone doesn't have an effect and we shouldn't be worried about it? That the only application is in conjunction with insulin? I'm asking that because I don't understand.
We're using it in conjunction with insulin because TZD is going to help us target what part of the pathway might be responsible.
Okay.
And if we find that the insulin TZD group shows less plaques and tangles over just the insulin, which should show less over just the control, then we can then suggest that there's a problem downstream of the PPAR gamma that's causing the pathology to happen. And we wouldn't be able to tell you what part of down there. We could just tell you that it's not this top half. Further research can now investigate.
Great answer. Great answer. Very good. Okay, only thing else I want to touch on base, I'm concerned that you guys have too much work cut out for yourselves. Not knowing what's involved in the laboratory for doing all these steps. It's a very multifaceted project. It has a lot of different moving parts. Clearly your group's going to have to segment into many different teams to tackle the different parts, which is the only way you can possibly. But each team's going to have to communicate to each other team, and each subset's going to have to communicate with each other subset. It's going to be a very complex communication pathway. A typical lab management software or lab management timekeeper is going to be inadequate, I think. Have you given thought of how you're going to do all the separate pieces, keep everybody informed, make sure that the left hand knows what the right hand, what the fifth hand and the eighth hand and the 12th hand, what you're all doing, and so that you have a cohesive understanding when you get to the end, and you have a good product because it wasn't a critical part in the middle that was jumbled up because of communication. How are you going to pull that off?
I think a significant portion of this semester, since we're not doing pilot testing until probably the very end, is going to be focused on like lining things up exactly and not jumping into things too quickly before we have the kind of infrastructure to deal with it. And I had another point and I forgot it.
One other thing that we had talked about before as a team, this is going sort of along with the organization but more with the consistency, was to have a specific group or person in charge of each of the separate data collection, like the facets of the data collection. That way there wasn't so much overlap, and also we wouldn't have to be trained in so many different things, each of us. We could focus on one, and then that study would be, that aspect of data collection would be carried out consistently by that person or team, like each time it was performed.
And that's important. That's a lesson you have to learn early on. Like, if, Caroline, you're in charge of some nostril squirting part of the lab, right? And Vinny's not, but at the end of the day, he's got to understand exactly what happened there. And he's got to understand what went right and what went wrong. So that communication's all, even though that's not your job, you're not the nose squirter guy. So, but you have to understand totally what it's all about.
Yeah.
So that communication is going to be very hard with this project and with, the team. So you have to make sure that you're really on top of that. It's going to be more difficult for you than some other teams.
My experience, especially with the rodents, theirs is more complicated than this.
Well, this sounds complicated.
Well, I think the complication is gonna be quite honest,
Sorry.
that, and we talked a little bit about this, you need 45 transgenic mice at about the same stage,
Let's leave that open and see what
which then makes it easy to go down, but what if you don't? So, that's something I guess we'll have to talk with Kara and Heather about, of almost splitting it, staggering some things.
That was a backup plan we had discussed
Yeah.
at the start.
I do think something you'll have to pay attention to, and Team Medics wrestled with this, and they were working with cells, so that was far less of a concern if someone treated their cells twice than if you do something with the animals twice, is making sure you stick to your lab schedule and that you all are keeping track of who's doing what, because you don't want someone to give your group 2 insulin twice, or your group 3 TZD and no insulin. So you need to make sure that you're communicating. So I know that you all have a lot of the SJs on your group, right? So someone might move some things around because they think it's better that way. You can't do that once you're in the lab. So y'all need to make sure that there's trust and good communication, and that's, you can learn from team medics, and luckily they just lost some cells. We don't want you to lose some mice, in that process.
Well, Kara's good at this, and I am, because we work with Adam as well. We have sheets for everything. Books for this, books for that. And initial line, you initial it, it was done.
That's good.
Okay, wanna go ahead and?
Yes, so how did you as a team explore, assess, and integrate scholarly resources into your proposal writing process?
We first started, we have used a lot of different types of sources. We've looked at patents with the help of Jim Miller, and to see like what drugs are out there now being used to study Alzheimer's disease. We've looked at a lot of journal articles, which any research team's going to look
Love
at.
journal. Oh, a lot of journal.
A lot of I'm sorry, I'll speak up. A lot of journal articles. We've also looked into books, watched videos of procedures, and a lot of different types of literature we've at least looked at and have integrated. And if someone wants to take it off and talk more about our research or types, I can keep going on this.
Well, we've also have great resources in our mentor and Dr. Kara and everything. So we've learned a lot from them, and whatever they've said, we've kind of like, gone back and took a deeper look into it through journal articles, like Ashley said. So that was also definitely something. I think we cited personal communication a couple of times in our paper.
A big part of it is also finding the holes in the literature, where no matter how many articles we look at, they're still not knowing this mechanism. They still haven't done this part yet, and and we're lucky in which it's there's still an unknown. It still hasn't been solved yet, so we can still get in there and make a little bit of.
Which one of the things you guys did that, if it was me and I'm doing a study, one of the things that I often look at to find out who else is doing it, or maybe get a little bit more meat of information. We did this in one day.
We looked at grants that were
Right.
being recently given out
Yeah.
to find out what the upcoming studies were gonna be based
Yeah.
on preliminary stuff that people shouldn't have done to get grants.
Right.
Why shouldn't it done?
Well, I mean, it's controversial if like people should like do a little bit of research to show that they like deserve money for like, I don't know, a whole grant.
Well, I wouldn't say it shouldn't have been controversial. I mean, seed data is what a lot of people consider. You do a small scale, sometimes inconclusive study that gives you direction. That's not negative. I mean, that's where a lot of big grants are sparked.
Well, and
A
I
lot
think I
of
told you
Gemstone
guys,
projects end up being
I think I told you guys this, it is very common for somebody to submit a grant proposal and say that they will do X, Y, and Z. Should I add another one to that? How about A, B, C, D, A, B, C, D? And they've already done A and B.
Right, that's standard procedure.
I've never done that.
Standard procedure.
I haven't, the grants I've gotten, I've never, this is it, man, take it or leave it.
Okay, you're a pure scientist.
And also, Caroline, I think you did a really good job explaining your Medicare cost figure, and I think you did a better job saying it than you all did writing it.
Uh-huh.
So you might want to go back to the video and take some of what you said and put that in. And also, Ashley talked about type 3 diabetes, and I think you made a connection in your presentation better than you did in your paper, so you should also consider adding some of that. And I think what became clear to me after your presentation and answering the questions, so you're looking at the pathology, not the treatment, and I think that needs to become more clear in the paper as well, because I think I was confused about that until the presentation. So if you're looking at the pathology,
The mechanism.
then I think that helps answer Dr. Cole's
Right, that's
question.
where my rate question came from.
And that helps clarify and helps you show what your contributions are to the literature. And also, I think maybe limits places where you can find some money too.
Speaking of money, your budget I think looks totally reasonable, and I think it's pretty realistic as far as I can tell. You got a lot of stuff for free, which is wonderful. This is a detail in the file that you sent the reviewers review, just like you put the grant agency review it. The columns on your budget were messed up, and it made no sense. I mean, you stared at it, and if I'm a proposal reviewer and I can't make heads or tails of the budget sheet, it's in the no pile. So that is a critical, just small little column width problem, which could have,
Yeah.
if this was a competitive process would have squashed your proposal. Just small detail. Other than that, okay, $8,700 you need to find from someplace. Where's it coming from?
Well, we just formed a grants committee, so we've been meeting and stuff. So and we found some places that we can apply to. So and we'll start meeting again soon for that. So
Okay.
grants are one.
Which ones? Because he's probably got a bombshell to drop.
Well, we were familiar with the Gemstone HHMI one,
Which
which,
no longer exists.
That's the
good.
bombshell.
there are two, I'm not sure of the name, I think one might be with the National Science Foundation.
There's one with the
Yeah.
National Science Foundation.
There's an NSF undergraduate project.
Yeah, the
Yeah.
undergraduate travel.
Yeah, I think there was one that we might have found through NIMH.
NIH.
Mental health.
Yeah.
Oh, okay, gotcha. Oh, that's good.
And then
Keep up this.
I'm not even-
we were confused as to whether like we could apply for grants under their like doctorates or
Oh.
whatever, or if it had to be as a student group.
Because a lot of them said like, oh, 10 years. We must have 10 years of experience with after.
The PI can be your mentor.
Yeah.
For most graduates, they want a faculty staff member as PI. Ones that are student specific, the student groups can be mentors, but they all probably want an advisor to sign off on it. Okay, that's great. I'm glad you're hustling because you're going to need to find some money. There's nothing in here on travel, conferences.
We're actually planning to attend, there's an online neuroscience conference which is headed by some people at NIH, which is in the middle of spring break, which has a lot of discussion on Alzheimer's and open forums where you can like go into different rooms and
Mhm.
stuff. So not that we would present, but we want to take part in that as like a little conference experience.
Just remember, if you're applying for grants anywhere, include in your budget money to travel on.
There's actually a travel grant we're considering.
Great. I mean, the more you can get out there and see and listen and learn and
Mhm.
present, all the better.
Yeah, it's unfortunate. You probably will not have data next November, I would assume. Which is a
Okay.
shame because, I think it's November, is the experimental biology deadline. Actually, it's October 18th usually. And EB, experimental biology, is in DC.
That's cheap travel.
Yeah.
Preliminary
We
results.
want to use some pilot study.
Not to that
I
meeting.
don't know, I don't know if they do
It's pretty high powered meeting.
Yeah.
But the point is, when you get a chance to create a budget for money, include your travel costs, of course, supporting your required research expenses, publication charges, those are all great things to put in there. All right, but good, I'm glad you're not waiting for HHMI to come through because of name.
It is the last year of ACC, IAC.
Yeah, we're trying to get money from other places now, but it's a slow process to have it. Okay,
Sucks.
one thing we want to do is make sure that we give you guys the chance to ask any questions you might have of us, programmatically, professionally, what you might do.
I get to see him Wednesday.
Okay, forget him. Any questions you might want to ask of us about anything that is related to your proposal? We all good?
Are we
I
gonna
feel
get
like
an
we're having
award?
questions later.
Do we get an award?
For this? We'll let you know at the end of this week. We'll have all the proposals finish up last month on Friday afternoon. And then the top 3 proposals get monetary awards.
How much?
Wait,
I
is this
think
the library one?
It's library award.
Are we at a disadvantage?
Because Natalina's not here.
Well, we're recorded.
No,
Oh,
this is what they're gonna review, the video. First place gets $2,000, second place gets $1,000, third place gets $500.
Money is money.
That's right, it helps. Any questions for us?
Is there anything we should be telling Natalina at this point that she should? Is she okay? She wished us luck tonight and said like let her know if she needs to do anything
Yeah,
else.
just ask her whether she likes chocolates or flowers, or ask her what she likes.
Thanks for
But you should incorporate all the feedback that you receive
Right.
today, and I have some edits that I'll send you. And work with Dr. B to make sure they're edits you want to accept. Most of mine are, you have 2 periods here, or you forgot a year, or something like that. But there may be pieces that we talked about using a different kind of proliferation technique instead of the BRDU, you wanna think about changing that now so that you're not scrambling in 2 years. So make as many updates as you can to your proposal at this point, you'll thank yourselves as seniors.
And along those lines, in the lit review section, the introductory section, I mean, it was well done, but there's pieces that kind of don't fit for one reason or another here or there. As you refine your knowledge and refine your thinking and refine how you want to present this, you're going to want to polish that, narrow it down, and clean it up so that you, as you know more what your outcome's going to look like, what your story is you want to tell, you want your introduction to focus in on that. So you're going to streamline that and clean it up a bit as you go. I mean, it's well done, but it's a little disjointed in some places, but that's almost to be expected. So just plan on cleaning it up as you go forward.
Did you all think that it was too long for a proposal? Because I know it was on the longer side.
We've seen longer.
Good.
We've seen longer. But word count doesn't matter. It's just, you want to get the message through and just make sure it's straight. It's a little bit wandery in some places, but it's common at this stage of the game.
There's also some places I put a note where you want to find the primary sources, and that would certainly help with the library award. Some pieces you cite are good, but I think if you dug in a little bit deeper, like Health and Human Services, they are probably citing research that had been done, so you can find the primary source.
I know at least for a couple of them, we had to go in and talk with our librarian because we couldn't find them, and she tried to help us. And for a few of them, she's like, if you really can't find it, leave them in. But I'm
Yeah.
sure there's some we can
Yeah.
dig a little deeper.
In the rule of thumb, most agency reports, there's fundamental
Yeah.
literature supporting them. So you need to dig down to that fundamental literature and not an agency report. They're usually just regurgitating what someone else has published. Okay, anything else?
Other
I want to
than,
thank you for a great review.
other than excellent. I
Yeah,
mean,
very good
I
job.
think
Well done, very well done.
your presentation now is gonna give you a good idea how to go back and change some of those things to get it, not focused, it is focused, but this whole concept idea of why you're using TZD, because it came really clear here. And that's gonna help you. But I love the slides, great, few words, visible.
And you had to teach your mice how to swim?
They know.
No, it's only, it's a small amount of water so that it's not like,
They're not going up
they
there?
shouldn't
drown.
as long as we're watching them,
They
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 won't
kind of do a
drown.
little paddle.
Like it
Yeah,
won't,
they-
I was like,
they-
it won't be, it won't be like,
It will have
That's
to
it.
give them
We definitely need a video of that.
behavioral.
it won't be.
Yeah, that's
I
why,
didn't do
that's
that.
why we've given them an entire week, because they will have training for the cognitive testing that precedes the actual testing, so that will span a week long.
They'll learn
And
how
they
to
will take a
swim.
whole week.
So they do get training, not necessarily in how to
Shannon's a
swim,
lifeguard.
but-
Lifeguards on
Yeah, I'm
Zoom.
a lifeguard.
Me too. I'm
Okay,
an
sit
ex-lifeguard.
here and watch me, okay?
I've watched more boring in a pool.
Oh, well done, you guys did a great job.
Yeah.
Yeah.
I see.
Yeah.
Well
Very
done.
good,
I've never
very
seen
good.
it. 7.
Where'd you? Same girl 4 times.